بسم الله الرحمن الرحيم به نستعين اللهم علمنا عنك يا رب فهمنا عنك يا رب. Fifth day, gene annotation and unlocking the secret of the genome. Today we'll delve into the fascinating world of gene annotation. A key process is understanding and interpreting the vast information encoded within our genome. What is gene annotation? Structural annotations and a function annotation. Structural and functional annotation. These type of annotation focus on identifying the physical layout of a gene within the DNA sequence, including where is the exon, introns, and the regulatory element. While functional annotation, it is going beyond the structure. This annotation is assigned the biological function of gene and the biological function of the products of this gene, revealing what those genes actually do in our body. What are the importance of our gene annotation? Number one, unlocking the gene function. In our disease research and finding a treatment, unveiling evolutionary history. Unlocking gene function, gene annotation help us understand how genes are regulated and what role they play in intricate machinery of life. Disease research and treatment. By understanding gene function, we can better study disease. When I know the genes that are normally expressed in this tissue, in this cell, and I notice the divergence in the disease state, and I can understand, number one, what are the genes that are deviated from normal during this disease, and according to this I can develop a target treatment. Unveiling the evolutionary history. Comparing annotated genome across species provide valuable insight into evolutionary relationship and how genes are changed over time. Essential tool for gene annotation include NCBI Genome Data Viewer, Ensemble, PROCA. NCBI Genome Data Viewer, this is user-friendly tool, allow you to explore annotated genome, searching for specific gene and their characters. Ensemble offer a wealth of information of gene annotation, including detailed visualization and functional prediction. PROCA, this is powerful software, is specifically designed for annotating bacterial genome, helping to identify genes and their function in these organisms. Decoding the language of genome. Step 1. We identify the coding and the non-coding region. Okay. The first step is to distinguish between region of DNA that code for protein, which is called coding region, and those that don't code for a protein and called non-coding region. Step 2. We locate the start and the stop codon. Okay? Within coding region, specific sequence called start codon and a specific sequence called stop codon make the beginning and the end of the gene, guiding those two codon. The first codon start the process, the stop codon stop the codon, so both codon guide the translation process. So the first step, I need to identify the coding and non-coding part. After that, I need to know where I start to transcribe and where to stop. Step number 3, we compare sequence with database. By comparing the identified sequence with database like BLAST and UniPort, we can predict the function of this gene and their protein product. From here, from knowing the coding part and the start and the stop, I can now know the sequence of nucleotide. Okay, this sequence can be for nucleotide alignment using BLAST. I can even translate this nucleotide sequence and to understand or to predict the protein that can be produced. Okay, so we can use these bases like BLAST and UniPort, okay, to predict the function of these genes through that sequence is related to this gene which is known function in our database, or this sequence will be translated to this protein which is already known, having known function in our database. This will be explained later on. What are type of gene annotation database? Ref sequence. So sorry, I have a pen. Okay. Reference sequence. UniPort GO database. Ref sequence, a comprehensive database of annotated genes and protein from a wide range of organisms, it offering curated and reliable information. UniPort, a vast resource for protein information, including functional description of this protein interaction with other protein, which is called protein-protein interaction, and link to other database. GO database, this database using hierarchical system to categorize a gene and the protein based on their function, which providing a standardized framework for understanding this gene rule. What the applications of gene annotation? What are? Number one, we can use it in personalized medicine. What is personalized medicine? I go to this person, I detect some genetic marker, I can predict through it its response to specific treatment, so I can tailor the treatment for him. Okay, gene annotation enable personalized medicine by tailoring treatment based on individual genetic profile. This lead to improve the outcome of this patient and minimize the side effect. Disease diagnosis by identifying disease associated genes. Annotation play a crucial role. So number one, I study the normal genes in this that is expressed in this tissue, in this cell, in the normal context. I studied the gene expressed in this cell or tissue when become diseased, so I identify the genes associated with the disease. Okay, let's go back to our database and know the function of these genes. Okay, so annotation will play a crucial role in accurate diagnosis and early intervention. Based on that, it will improve the process of drug discovery. I understand well right now that these genes are disturbed in this disease, so I will design drugs that target these genes or their product or improve the process, the biological process that is deviated from normal in this disease. Annotation help in identifying potential drug target, paving the way for development of a new and effective therapy. Agriculture biotechnology. Annotation facilitates the development of genetically modified crop with enhanced trait and improve food security and sustainability.